Hello, I'm Ophelia Becker, and I'll be talking about Dr. Richard Axel, MD, today as part of my biology role model project. I want to start out by talking about his early life. He grew up in Brooklyn, New York, to immigrant parents who were relatively poor, had absolutely no interest in science, and absolutely loved basketball. Ended up getting transferred to a high school for intellectually advanced students, where he was on the worst basketball team in New York City, which was very unfortunate for him personally. But also going to that high school led to him developing this relationship to the arts, where he went to opera shows all the time, loved classical and modern art, loved theater, loved Shakespeare, and ultimately loved literature, which led to him getting an undergraduate degree at Columbia for literature. He got a BA specifically, but also, well, in his time at Columbia, he had to work to support himself because he didn't have that financial support from his parents. So he ended up working in a glassware or cleaning glassware in a lab. And then he took an interest in the material that was being covered in the lab and got fired from the glassware washing position just to be rehired as a research assistant. And so then was kind of mentored in science at the same time that he was receiving his BA. And then something really unfortunate happened after he graduated. The Vietnam War kind of took over. So there was a draft, and he really wanted to be doing research. So he ended up going to medical school instead at Johns Hopkins. And he was really bad at everything there. And so he kind of got like taken under the wing of some people there who allowed him to graduate early, as under the condition that he would never practice on live or even dead patients. And then later on, he was also a postdoctoral fellow at the National Institute of Health. Primarily, his areas of study are molecular biology and neuroscience. He's a self-described neurobiologist. So then his kind of initial foray into research would have been this stop over in Saul Spiegelman's lab, where he worked on the development of a synthesis technique for RNA in vitro. And then this also led to some gene transfer techniques where any gene could be basically transferred from any cell to another cell. And then also led to some important developments in HIV and AIDS. So eventually he began to realize that this genetic work that he had been doing could also be applied to the brain. And so that's where we get into this whole olfactory sensing deal. So in leading up to 1991, him and his colleague Linda Buck were doing a lot of research on olfactory perception. So they isolated 18 members from a multigene family that code for proteins that function as receptors in cells, and they are exclusive to the tissue inside of the nose. And so those gene family members are what are likely responsible for coding for diverse odorant receptors. So they kept finding more and more of these receptors, eventually coding for 1,000 plus genes that are olfactory receptors. And all of this research led to both him and Linda Buck receiving the Nobel Prize in Medicine and Physiology in 2004. So where do you go from there? Where did the Axel lab go from there? Well, currently their self-described focus is what are the spatial and temporal scales of brain-wide neuronal activity, which seemingly has nothing to do with olfactory sensing, but I'll talk about that more later. But also some other questions they are asking is what patterns in neural activity elicit a behavior? How do you relate the external world and innate senses? And then how is value derived from neutral neural stimuli? Because of course, neural stimuli is basically just electrical impulses, which have no inherent value associated with them. So how are living creatures from humans to flies incorporating those values and then translating that stimulus into behavior? Really, that's the kind of key point here. Okay, so in this paper that his lab, the Axel lab, put out in 2023, they had this specific figure, which essentially is showing that the more vigorous activities that flies engage with, like flailing or running, leads to increased brain activation. And so to generate a lot of this data, they were using this technique called escape microscopy, which stands for swept confocally aligned planar excitation, which sounds complicated, but realistically, it's just a high-speed 3D imaging technique for live organisms, such as the flies that were imaged here. And basically, it's just a light that sweeps and scans across the sample, and then that light passes through the sample and is received by a descanning plate, and that translates into a 3D image. It is a more complex process than that. That's like kind of the simplified version. So then data from those 3D images can be gathered and then transformed into representations like this. So let's look at A and F here first. They're called delta FIF, where in figure F here is Z fitted, and then the left shows running, and the right shows flailing in flies. And then we also have B, C, and H in this figure here. They're all regression weights. So this first one is for back grooming versus running. C is for back grooming versus front grooming. And then H is for flailing versus running. And basically what they're showing here is cell activation. So the gray spots on these graphs are showing where cells aren't activated. And then the red, orange, and yellow spots are where they are activated, and the colors correlate to for what duration of time. And so you can see pretty clearly here that there is a marked increase in the amount of activation when you're looking at running and flailing, which are considered more vigorous movements. And then there's also D, which is kind of the entirety of this image here in the middle plane. And really, like, the gold part is showing which cells are activating, the blue showing how much they're activating. So that's the gist of that. And then there's E down here, which is a relative rate of variance, which is normalized to running for all flies and all cells used in the experiments. And it's just kind of a way of really having a simple image to represent some of this information. And then lastly, here we have G in this figure, and it's showing the activity from 4 sample neurons, and then it has a regression of the information shown in F here overlaid, and then it also has the behavioral state shown below, whether it's running or flailing. And yeah, so like I said, this all just really shows that vigorous movement in flies leads to increased activation in the brain. And so then I assume personally that this lab is hoping to continue down the path that they have been of really understanding how you can look at behavior based off of brain activation and like neuron activation. And then I'm assuming that they are hoping to eventually integrate that into their large body of research into olfactory receptors. So then really the whole reason I did this project is because I'm identifying Richard Axel as my biology role model. So why is he? And I just like the biggest thing for me was just hearing how he talks about his peers. He really gives them respect from his undergraduate students to his graduate students and even to his own personal mentors. He's always acknowledging people, always directly specifying names. And also like he was so humble in the way that he accepted his Nobel Prize, where he said, it's not about me. It's about my research and all of the people that contributed to my research, which I just thought was very respectable. And then he also had this modest upbringing and integration of art and science, which really applies to my personal background. So it's really cool to see somebody who shares that. And he has this great sense of humor and humility whenever he is giving lectures, and I really appreciate that. So yeah, thank you for listening to my talk about Richard Axel. Here are my references.